Good evening, I'm Jeff Miner, President of the ASMB. It's a great pleasure to introduce Dr. Hans-Peter Bächinger, winner of ASMB's Senior Investigator Award, which is presented biennially to an established researcher for outstanding continued contributions to the field. Hans-Peter received his PhD from the Biocentrum in Basel, Switzerland, and then spent some time at the Max Planck Institute in Martinsried, Germany, before venturing to UCLA for a postdoctoral fellowship. During this period of training, Hans-Peter was trained by pioneering giants of the matrix biology field: Jürgen Engel, Klaus Kuhn, Rupert Temple, and John and Lisa Fessler. In 1984, he joined the faculty at Shriners Hospital and Oregon Health and Science University in Portland, where he remained for the rest of his illustrious career. Hans-Peter made foundational discoveries about the structure, biosynthesis, and folding mechanisms of collagens that he will tell us about today, including the discovery of the family of perolyl-3 hydroxylases. His discoveries are particularly relevant to our understanding of how mutations in various collagen types cause a diverse range of diseases, including osteogenesis imperfecta, Ehlers-Danlos syndrome, chondrodysplasia, epidermolysis bullosa, Gould syndrome, and my personal favorite, Alport syndrome. Congratulations, Hans-Peter.
Thank you very much, Jeff, for this very kind introduction. My title today is Mysteries of the Collagen Triple Helix, and I will just go through some of the steps that actually Jeff already mentioned. I started my thesis in Jürgen Engel's lab, and protein folding was really a big problem at that time, and so I thought, oh, this is easy. And what I had to work with was peptide extracted, type 1 collagen from synthetic XY peptides. And it turns out the easy part was nowhere to be seen. We had type 1 collagen, native, and we could never refold it. We had partial refolding, and that was it. The synthetic peptides didn't really help us either. So the breakthrough came when Rupert Temple extracted the amino terminal propeptide of type 3 collagen. This allowed us to see a fully folding, unfolding reaction. And later on, he also gave us peptide extracted type 3 collagen. So with the amino terminal propeptide of type 3 collagen, we actually could show that if you have native material, you unfold it, and you fold it back very fast, you can see there is a fast phase and a slow phase. If you wait for a while, you can see that the fast phase is much less available, and the slow phase is highly increased. If you plot the amplitudes, you actually get the rate of trans to cis isomerization. So collagen contains a lot of proline residues that have a significant amount of cis peptide bond. All the other amino acids basically are equal trans. So this was our first breakthrough that we had. And when we looked at it, that type 3 collagen refolding, which was also the first full-length collagen that we could basically 100% refold, we saw a very, very strange kinetic, which made perfect sense for a simple-like folding with cis-trans isomerization as the rate-limiting step. So this is highly unusual to see a straight line in protein refolding. Normally you have a fast phase and a slow phase of folding. So then I went to John Festel's lab to see if the theory behind our in vitro studies had any significance, because the cis-trans isomerization, if you have a newly synthesized protein, all the peptide bonds are actually synthesized in trans. So if you fold the protein right after synthesis, it doesn't matter if there is a cis or trans isomerization going on. And at UCLA, I also met our next speaker, and Lynn told me I'm absolutely not allowed to talk about random collagen, so I won't do that. Anyway, we looked at the biosynthesis of type 3 collagen in chick embryo blood vessels. And what we found is that the theory that folding occurs directly after synthesis is wrong. We could clearly isolate monomeric chains, and we could pulse chase these monomeric chains into trimers that were disulfide linked at the carboxy terminal, and then we could isolate trimers at a later stage in the process that are disulfide linked at both ends. So clearly, the cis-trans isomerization rate is an important aspect. And in 1984, there is this Günther Fischer, who I became friends with, worked in East Germany and published a paper where he extracted an enzyme from pig kidney that actually catalyzed cis-trans isomerization. I read this paper and I said, oh, I must do that. I mean, we just established a mechanism. This is going to be very important. And so we isolated the enzyme. I wrote a grant. And the NIH said, nah, this enzyme doesn't exist. They said it comes from an obscure journal, and the buffer itself can catalyze this. So it took quite a while, and finally, they published that paper, and at the same time, it was actually shown that the pig kidney extract that we used was cyclophilin B. So that was exciting. So this led to a whole additional studies that we used chick embryo fibroblast, because we now knew it's cyclophilin B. We could use cyclosporin A as an inhibitor. And as you can clearly see, synthesis in the pulse chase is really delayed. And we knew that FP506 was an inhibitor of FKPs, and the partial inhibition of FKP65 that also inhibits the rate of biosynthesis. So you can ask the question, I mean, collagen triple helix is a very simple structure. So what could possibly go wrong in a process like that? Well, it turns out there is a lot. So we have all the mutations in the different collagen types, and you're all familiar with them, so I don't have to go through that. So, but is there a possibility that there is mutations in the whole folding machine? And over the years, we developed what Yoshi calls the molecular ensemble of collagen biosynthesis. And it does involve an enormous amount of different proteins. This is just, it's probably way outdated now. So we have to fold, we have to secrete, and we have to modify residues in the collagen triple helix. So the folding machinery is very complex. There's many stable and transient protein complexes that exist, and there is multiple interactions. So many of the molecules not directly associated with collagen biosynthesis so far, and it's an older slide, so we now know that there is many of them are important in connective tissue diseases, and the mutations in the folding machinery might sort of giving overlapping phenotypes. So what we try to do is look at the post-translational modifications, which is proline 4 hydroxylation, proline 3 hydroxylation, lysyl hydroxylation, and no glycosylation. We have to deal with proteolytic cleavage, oxidation and cross-linking, and then all the molecular chaperones, which is general chaperones, disulfide isomerase, disulfide reductase, oxidoreductase, the IAs, like the binding transport translocation, degradation, disaggregation. So there is a lot of questions how you study such a complex system. So we know there are stable complexes, P4H, PI, obviously P3HCl perhaps, and often many others, but there is also transient complexes. And the technology to decide what is a transient complex and what is a temporary complex is not that easy to do. So when we look at enzyme reactions, we can either use Michaelis-Menten analysis or Briggs-Hawking analysis, which in many, many cases just work perfectly fine. However, in the collagen biosynthesis, it's much more complicated because the modifying enzymes require only unfolded chains, or many of them do only unfolded chains. And so the substrate availability is going to be time dependent. And then in addition to this time dependence is that the rate of triple helix formation is tightly regulated by PPIases. So we can ask what of the biochemical changes cause disease. So lack of 3' could lead to different collagen protein interactions. We've shown that a lack of type 4 3' leads to a decrease in nitrogen 4 interaction. It could increase glycosylation, disturb fiber assembly or domineralization, and the altered matrix could feed back and lead to difference in cellular response. And so that could be like fibrillin and morphin disorders. So we can ask what are the consequences of glycine mutations on the triple helix. So there's a local structural change, there's lower stability, slower rate of folding. And obviously these mutations can interfere with collagen protein interactions. So we studied just an example, barely visible, but there is actually an alanine glycine mutation in there. Clearly doesn't disturb the structure much. Or we could have a sugar attached, which sort of gives you an additional element of structure. And what we then looked at was EDS type 6, because there was clearly 2 different mutations in the alpha-1 channel, type 3 collagen, and there were fibroblasts available. So we thought, let's try to study this in a quarter fragment. So we generated trimers with the mutation, and we did glycine 910 alanine and glycine 910 valine, glycine 1018 alanine, 1018 valine, and we looked at how the refolding kinetics changed due to this mutation. I have to tell you right away, these studies were not very good because the mutation is in all 3 chains, which is not what occurs in nature. But what was very obvious was that, first of all, the size of the mutation plays a role, and that the rate changes exactly where the mutation is located. And you can see different effects. Alanine is not that bad, valine, very bad. So this told us that we are on the right track. So the folding is normal until you hit the size of the mutation in this simple-like folding, and the slow step has to occur after the mutation because you have to get back into register. So it obviously depends on the size of the amino acid that replaces glycine, and the rate, the slow step, is 2 to 10 times slower than the normal folding. So, and it depends on the sequence where you basically have to renucleate. So people thought, well, let's do some chemical chaperones, and there was 2 entities. And my take is that you can't really use chemical chaperones. I mean, you need high concentrations. Yes, they do have effect on cells, but this is not a solution that could be applied to a patient. So what we were thinking is, well, why not use chemical pharmacological chaperones? And so we started out very simply with a few hundred compounds or 1,000 compounds, and we didn't even test all of them. But what we did, we developed this assay. It was the mutation G910 valine. We used this quarter fragment, and what we could see was that certain compounds like 11C actually increase the rate of folding through the mutation. So this was encouraging, and we had some concentration dependence. And so we started thinking, well, there is hundreds of thousands or 500,000 compounds that need to be tested. So we actually developed a throughput assay, and I don't want to really bore you with that. But then came a crucial experiment from Sergei. We used the NC2 domain of type 9 collagen, which allows us to make heterotrimers of any combination. So this is a huge breakthrough, and basically we had to start over because we had homotrimers with 3 mutations. This now allows us to exactly pinpoint the mutation. And what it looks like is that you use these 3 peptides from type NC2 domain of type 9, and you attach whatever sequence you want. And you could actually verify that the type 1 stagger is alpha 1, alpha 2. So this allows us now to completely make pieces of collagen triple helix with the right stagger, with the right mutation in the right place. So if we can go through with the assay and find active components, we then test them in CD for folding, test refolding cells with pulse-chase experiments, and we could test them in mice. And the idea was to screen large libraries of 500,000 compounds or more. So, this is just a list of active compounds measured on the old method. So I wouldn't put much value in that. So when you start thinking about pharmacological chaperones, you can think of about 3 classes. Some are good, some are not so good. So unselective, it just increases the overall folding rate. Positional are very interesting because they might actually help renucleating the collagen sequence. Universal ones that can accelerate the nucleation. So there is a lot of possibilities, and we're not quite sure what we're actually going to look for. So the approach will clearly not work for mutations that affect a triple helix protein interaction. That has nothing to do with the folding. It might be helpful for less severe mutations because we could inhibit the increase in glycosylation, the increase in hydroxylation. So that's possible. Another thing I want to mention is that many more tissues are actually affected when we looked at mouse models and looked at them in terms of the human disease. We see many more tissues that are actually affected in the mouse than are described in humans. And obviously, overlapping phenotypes are observed in mouse models of negative tissue diseases, especially true for mutations that are not collagen, but in the molecular enzyme. So I want to just very quickly get to the warm blood vital form syndrome type 1, and hopefully Yoshi will find the time to summarize the results and publish them. These are in mid-sized horses originating in Europe, and they have extreme spleen fertility, and joints are lax and hyperextensible, and the affected foals, they are all euthanized. So this is for homozygous mutations. And it's a homozygous mutation in the gene for LH1. it's glycine 678 arginine. And Yoshi did quite a few experiments. You can see that proline hydroxylation is completely normal, but you have a lot of hydroxylase versus hydroxylase. So hydroxylase. So why is this important? We looked at the diet, by arch rotation, nothing really. However, the consequences are dire, and this is not proven. But Benamina, that discovered this mutation, actually finds it in all the famous racehorses. So, name a horse, I can basically assure you it's a head in this LH1 mutation. And the head normally, if you just keep them, there is nothing wrong with them. And the problem is that if you start racing these horses and put them under stress, you might induce all sorts of problems. And for a while, it was my hobby to look, oh yeah, that's all died. I went through the bloodline. Oh yeah, it's a carrier. So I think that this is something that could be addressed and could potentially save the horse racing industry. And it's very controversial, but I think it's something that needs to be done. So I've blamed Yoshi now for not summarizing his results. So I have another claim to dish out that is too late. I have now known for 8 years that fibrillin 1 contains 3-hydroxyproline. And wouldn't that be interesting to know? I would. So I would like to thank, obviously, all the members that worked in my lab, my graduate students, my postdocs, and it has been a pleasure to collaborate with many, many of you. And again, I want to thank the ASMB for giving me this chance. Thank you.